Hi, so this is the first video of this week. We're going to be completing the protein synthesis topic, and we've two parts to it. We've got mutations and chromosome mutations, and we've also got all the question pack to finish off too. So let's get going. Okay, so we are working our way through the PowerPoint number 3 for this topic, which starts with a bit of a recap of what we did last week. So the processes of transcription and translation. So I would like you to pause in a second, copy down and complete the table by placing ticks and crosses in the columns. Okay, so pause. Right, so if you're coming back now, you should have completed this task. So we should have our completed table. Okay, and we can check against this one. So transcription uses tRNA, translation uses it, so we've got to tick the right one, okay, all the way through. So uses tRNA, of course, transcription doesn't, but translation does. Happens in the nucleus. Transcription happens in the nucleus, obviously for eukaryotic cells. For prokaryotic cells, because they don't have a nucleus, it just happens in the cytoplasm. Okay, but transcription for eukaryotic cells anyway happens in the nucleus, whereas translation does not. Uses RNA polymerase, so that's the enzyme that joins our RNA nucleotides together to form the mRNA, which happened in transcription. It's not involved in translation at all. Happens at the ribosome. So that is, of course, translation. And finally, it involves the formation of peptide bonds. That's just in translation as well. So I hope you have got all of those correct. So we've got a little bit of a quiz now. Again, just recapping what we did last week. Okay. So could I ask you to keep pausing, having a go at answering the questions and get all the task for the quiz, and then starting the playing again to see if you've got the answer correct. Okay, so first one, where does translation take place? For this one, you should be quite quick off the mark, so you just stay with me. Well, I tried to be a little bit tricksy here. It's on the ribosome, and it depends on the cell type whether or not you're going to say on the rough endoplasmic reticulum, if you like, on those ribosomes, because that could only be possible in a eukaryotic cell. So it's always on the ribosome, that's always true. For a prokaryotic cell, that ribosome would be free in the cytoplasm. So I think just stick with A as the true answer. Which of the following is not involved in translation? Oh, we should be fast on this one then. Just DNA. So DNA is not involved in translation at all. What are removed from the pre-RNA at the end of transcription? So what do we know gets removed? It's the introns, or otherwise known as the non-coding DNA. So there's the sections within a gene that do not code for the amino acid sequence of the polypeptide. Okay, we're going to meet variable number tandem repeats next year. These are also non-coding DNA, but that non-coding DNA is found between genes. Okay. So it's just the introns that are removed. And of course, the name of the process where that happens is splicing. So that was missing a little bit from our gap fill task. So make sure you've got the key term splicing there. Okay, what anticodon would be found on the tRNA which joins to the codon of UAU on an mRNA? Which one would it be? You're looking for a complementary AUA, as the anticodon on the tRNA is complementary to the codon in the mRNA. Okay, may need to pause me, give yourself enough thinking time for this one. Which is the correct mRNA sequence produced from this DNA template strand? Okay, I'm going to show the answer now. Okay, so C was complementary, but in RNA, so it's got uracil in there. So well done if you got that one. Okay, so we have done a bit of a recap. We're moving on today to the genetic code and mutation. Okay, so in your keynotes, you will find this list. Okay, so these are the features of the genetic code. Okay, I'm going to talk you through each one. You've got a little definition in your keynotes as well. Okay, but you can take additional notes if you want now on paper in front of you. So the first feature of the genetic code is that it's a triplet code. Okay, a triplet code. What does that mean? Sometimes the examiner asks for 1 mark. So this means that 3 bases within the DNA code for 1 amino acid in the polypeptide chain. Okay, second feature. This one's a little bit more difficult to define. Okay, it's a non-overlapping code. And the diagram on the right-hand side is to show that. Okay, but when you're asked to explain what is meant by non-overlapping, it's best to think about, for clarity, a description which means that each base is only read once within a codon. Okay, so if you have a look at the example in the diagram, if we were given that piece of mRNA, evidence that it's a non-overlapping code would be that there are only 3 amino acids coded for. Because if it was read in an overlapping way, like shown in the diagram, where the first 3 bases coded for the first amino acid, and then you just kind of moved up one base, so you read these 2 last bases again as the beginning of the next triplet that you read, and then the same can you see with the third reading. If it was read like that, of course, this strand of mRNA would code for more than 3 amino acids. So it is not, it doesn't get read in an overlapping code like that. It's a non-overlapping code like the one at the bottom. Okay, so each base is only read once within 1 codon. Okay, now the most interesting feature, I think, of the genetic code is its degenerate nature. That's the 3rd bullet point down. Okay, now to understand that, in your keynotes, you need to go to the codon table. Okay. Okay, so in your keynotes, you'll find a table like this. Okay, so this is a codon table, and it's what we use as scientists when we're trying to decode an mRNA molecule. Okay, so if we know the base sequence of an mRNA molecule, we can use this table to work out what the amino acid sequence is. So let's imagine the first codon that was on this mRNA was AUG. Okay, this one's a bit of an unusual one. If it was AUG on the mRNA, we'd go A, U, and then we're reading in this box here. And if you look here, G would be the last amino acid, so it's MET. So that would be the amino acid coded for. Now MET is a bit unusual. It only has 1 codon that codes for that particular amino acid. But have a look to the rest of the code. So for example, let's go just below to Val. Okay, so this amino acid has got 1, 2, 3, 4 different codon codes which code for the same amino acid. Okay, so GUU, GUC, GUA and GUG all would result in a tRNA with the anticodon complementary to the appropriate code, but all of them would bring the same amino acid. Okay, so how do you explain that to the examiner? Okay, so this degenerate code. Okay, so some of you will have worked out if it's a triplet code and we've got 4 bases read in 3s, that actually gives us 64 different triplet codes. And there are only around about 20 amino acids to be coded for, which means it's degenerate. That most amino acids are coded for by more than one codon. Okay, and usually we'll be asked for evidence, and we'll see an example of that in a minute. Okay, the next feature, okay, of the genetic code is that it's universal. That means all living organisms use the same code. So if an mRNA has got AUG as its codon, doesn't matter whether it's a bacteria, a human, a lion, on the ribosome, a tRNA with a complementary anticodon to that AUG will bring the same amino acid met no matter what species. Which is very cool, as we'll see next year, because, and as you already know, it means we can chop pieces of DNA from one organism, so we can cut a gene out from one organism and place it into another organism during genetic engineering now. And because it's a universal code, the organism that receives the new gene, when it expresses that gene, when it transcribes and translates it, it produces the same protein the gene codes for in the original species. Okay. The next one is not always something that you would recall as a feature of the genetic code, but I always think it's important to point out here that some of the codes don't code for amino acids at all. Some of those 64 different codes code for stop, which is the end of that translation process, as we learned last week. Okay, so I'd like you now to get your question pack. Okay, and in your question pack we're going to work through question 1. Okay, so we're going to work through question 1 together. Question 1A. mRNA may be described as a polymer. Okay, explain why. Now you can either pause as we've read through the question and write your own answer, or you can wait to listen to my answer and then record your own answer. Depends what level of challenge you want. Okay, it'd be more difficult to have a go yourself and then check your answers. Okay, so I'm going to put the answer up now. Okay, so it's made up of many nucleotides. That's the evidence we describe it as a polymer, because these nucleotides are, of course, monomer units. The same or similar units joined, many of them joined together by condensation reaction. Okay, you might push now for more detail there. Remember, our new spec, sometimes the examiner wants a little bit more. Okay, so this is an old spec question. So maybe add to that many nucleotide, include the word monomers, and you might even say joined together by condensation reaction. Okay, so the diagram shows part of an mRNA molecule. Okay, one thing you will have to get used to is the terminology that we use when describing the 3 bases that code for an amino acid on the mRNA. Of course, we call those a codon. Okay, when we're talking about the same 3 bases on the DNA molecule, so the ones that were transcribed to produce this mRNA, So it would have been a G, C and a T on the DNA. We call that a triplet, just so you don't get confused and use those keywords in the wrong way. So when we're talking about RNA, we use the key term codon. So this mRNA molecule has 9 bases in length. Can't count. 9 bases in length. And that means it's 3 codons long and 3 amino acids are coded for by that length of RNA. So this is really common now. So this is the kind of question you'll be asked a lot. The genetic code is non-overlapping and degenerate. Use their diagrams. You have to use evidence from the diagram to explain what is meant by, first, 1 mark for non-overlapping, and second, 2 marks for degenerate. Okay, if you want to have a go yourself, pause now. I'm going to go through the answers. Okay, so non-overlapping. Now, remember, you've got to use this information in the diagram. So the same base is only part of the code for 1 amino acid. Okay, and a good example here would be to use this first one here. Okay, and you don't have to use that particular one. You could have used another example, but here the examiner said, for example, the A in the code for arginine is not used in the code for lysine. So each base is only part of the code or the codon for 1 amino acid. It's only read as part of one single codon for 1 amino acid. So you write this in different ways, but it's got to be equivalent to that. Degenerate, much easier. Always the first mark point is the same. More than 1 code for 1 amino acid. And the evidence is up here, CGA and CGC both code for arginine. So 2 marks. So that's the most common way you'll be assessed on those 2 features. Okay, and it's usually evidence, so you usually have to give an example like that. Okay, so carrying on then, the table shows the percentage of different bases in 2 molecules of mRNA obtained from the same cell. Molecule X was transcribed from the DNA in 1 chromosome. Molecule Y was transcribed from the DNA in a different chromosome. Name base Z. Well, remember we're in RNA, therefore it's uracil. Complete the table by writing the percentages of base Z for both of these molecules. Remember, this is a single-stranded molecule, okay, but because these are percentages, if we know the percentage of adenine, cytosine and guanine, what's left to take us up to 100 would be our uracil. So that's the answers you should have in there. The percentages of bases in molecule X are different from the percentage of bases in molecule Y. Explain how transcription from DNA causes this difference. Okay, so some students find this one a bit tricky to word. So have a go if you haven't already, and I'm going to go through the answer. Okay, so the reason they're going to have different percentages of bases is because they're transcribes or transcripts from different genes. Okay, and those different genes will have different base sequences. So different parts of the DNA, different genes will have different base sequences. The mRNA molecules, X and Y, are complementary to these different base sequences, and so they contain different percentages of bases. Simple. Okay. Okay, moving on to question 2. Question 2 is a good question. Okay, so we're going to have a go at this one as well, because this is just our genetic code and applying it. So the table shows some mRNA-based sequences and amino acid sequences that would be produced from them. Complete the table to show the sequence of amino acids produced from the final mRNA-based sequence. Okay, 1 mark. So for this one, you have to look for where that has been coded for somewhere else to decipher it. Okay, so having a look through, we can see here this second one and that one have the same codon, so they're going to code for the same amino acid. So you do that for each one, sussing out where you've seen it before, and then you should get that. Okay, so sorry, that should have an R on the end of it as well. Use the information in the table to give one sequence of DNA bases that code for SIR. So there's, we've got our SIR. Now, this is of course the mRNA. So we're looking for the complementary triplet that will be in DNA. And then one anticodon for the amino acid. So that would be the complementary anticodon. And you've got a choice. No, you've not. They're all the same, haven't you? Okay. So it's going to be TCA, complementary, but in DNA, and then UCA, again complementary, but in a tRNA molecule it would be uracil. Okay, now same questions again, but what I'm trying to show you is that these questions are built around providing evidence. So they're presented in pretty different ways each time. Somehow the examiner will give you information about bases coding for amino acids. Okay. They used to give that codon table that I showed you earlier, but they've moved away from that because it was too easy for students to learn how to use it and sort of learn a rote answer. So they really want you to show that you're thinking about these as like a problem solving exercise. Okay, so evidence from the table that it's non-overlapping, evidence from the table that it's degenerate. Do you want to have a go and then go through the answers with me? Okay, I'm going through the answers now. Okay, so the first one, it's non-overlapping. So this is probably the clearest mark scheme. Okay, so if you want a really good answer, this first line's a really clear way of saying it. A base from one triplet cannot be used in an adjacent triplet. Okay. And the evidence then is using one specified one that you've got. Say for example, this one's a good one to go for. Okay, so it contains AGU. Okay, if it was read in a different way, so if it was read GUA UAG, then you wouldn't just have SIR. There'd be other amino acids coded for as well. Okay. Or if you picked that one again, we could pick this one here. Okay, so if this one was read in an overlapping way, not only would you have CAA, then you'd have AAC, and we know AAC codes for ASP. So you could say that because you haven't got that ASP in the amino acid sequence, that's evidence that it's only read in a non-overlapping way, where one base is only ever read within one codon or one triplet. Okay. Or you could have gone for an easier piece of evidence. All the peptides contain 5 amino acids. If it had been read in an overlapping way, they would have coded for peptides. Those sequences would have coded for peptides longer in length than just 5. Oh, so the marker scheme is there, but the answer that you've written might be a little bit, different, because there's lots of ways of evidencing from the table there. For degenerate, okay, it's not as varied, the answer. Some amino acids are coded for by more than one codon. That's your first mark point. That's always going to be a first mark point. And then in this example, we've only been given one, which is ACC and ACA, both coded for for thioline. Can't say it, I don't mind. So because we had those two different bases, two different codons, ACC, ACA. So that one was a bit clearer, less variety in the answers. Okay. Right, going back into your keynotes now, and we're in the mutation section. So we've just got to have a think about what mutations are. Okay, so most of the rest of this PowerPoint is really focusing on what mutations are. So from your keynotes, a mutation results in the change in a base sequence within a DNA molecule. It's really important that you remember that this happened during the process of DNA replication. Okay, so that's when mutations happen. When we did our DNA replication topic, you'll remember there were some questions where there were chemicals that were similar in shape to nucleotides, and they got in the way of the nucleotides during the process of DNA replication. And we said that they may stop DNA replication. Sometimes chemicals can just prevent one nucleotide from being added at a position, rather than inhibiting the whole process. So those kind of chemicals will increase the chance of mutations happening if they're in the nucleus during DNA replication. There are other things as well that increase the chance of mutation, but I think a lot of what we're going to be thinking about today is what the consequence of mutation is rather than how it happens. So this idea that if you have got to change the base sequence in the DNA molecule, when the cell expresses that gene, so following transcription and translation, that change may result in a different amino acid sequence. Now the may's in capitals, and that's because of the degenerate nature of the code. A change in base sequence doesn't always change the polypeptide chain. Okay, so where we have got amino acids coded for by more than one codon, sometimes a mutation can happen, which we refer to as a silent mutation, because it has no impact on the final primary structure of the protein. It doesn't change the sequence of amino acids in the polypeptide because it still codes for the same amino acid, even though there has been a change in base sequence. Okay, but if the primary structure is changed, okay, that might mean that the protein doesn't function anymore. I'll just go back to that and just think for 1 minute. It might not function anymore, but sometimes we focus a bit too much on this in this section. And I will remind you that you've talked about mutation in natural selection in GCSE. So although mutation we're looking at might cause a protein to be coded for that has got a different primary structure, and that might mean that the protein doesn't work. This is what causes quite a lot of genetic disease, non-functioning proteins. Equally, a mutation might be of benefit to an organism. So we'll come back to that idea later on, once we've got our head around mutation a bit more. Okay, so this is a section of DNA which codes, is part of a protein, part of the coding information, the exon of a protein, of a gene. And this is without any mutation. So we know this is the template strand. We use that to build our RNA. So this is our transcript. And then that is used in translation. Okay, so in the translation process, we know that this codon would be bound to by a tRNA carrying this amino acid at that first position. This next codon, CCC, we'd have a tRNA which would have a complementary anticodon to CCC carrying this amino acid, and when they come next to each other, they join by condensation reaction. Enzymes would join them together and a peptide bond forms. So now we can kind of skip all the detail and kind of summarize in a diagram like this our mRNA code and the polypeptide chain it codes for underneath. So without any mutation, we have met, pro, loose, so like that. And we always give the first three letters of an amino acid because there's 20-ish of them. It just makes, well, some of them have got similar names. If we give the first three letters, that's enough to discriminate between them all without having to write the whole names out. Okay, so let's think about a mutation. Oh, sorry, no, we won't. I'm just showing you again on your table. Okay, so when we did AUG, how you used your table to look up what that coded for. So AU takes you to this square, but then G takes you within that square to the bottom there. Okay, so thinking about CCC, where would our CCC be? So C, C, and then going to there, C, and then we've got UUA, so UUA, well, there we go, and finally then for our SIR we've got AGC. Okay, so that's how they were all looked up. Okay, so you can do that with any mRNA. As long as you know the codons, you can use this codon table to work out what amino acids are coded for. Okay, so now we're going to have a think about mutation. Now, in your keynotes, okay, so in your keynotes, you are working on page 11 at the top. Okay, so just a bit of a recap of what we've already talked about. So this idea, gene mutations involve a change in the base sequence of a DNA molecule, so of a chromosome. They happen during DNA replication. Okay. I'd underline this word spontaneously as well, because sometimes students have a misconception that you have to have something causing a mutation, something like an X-ray or something like that. But actually, gene mutations happen spontaneously as natural events, and as I alluded to earlier, are really important in natural selection, because you need a mutation to give you the variation in a population to be selected for. So we will come back to the idea of gene mutations that are of benefit to organisms in a later topic this year. Okay, so a mutation in DNA may result in a change in the sequence of amino acids in a polypeptide chain and may alter the function. So in that statement there's a lot of mays, okay? So let's just remind you that that's because of this degenerate nature of the code. So, you know, the idea that leucine's coded for by 6 different codons, yeah, so you can see leucine 6 times here. It means that if we were to change the codon from CUU to CUC, it still codes for the same amino acid. So some mutations we could change the codon code, but we will still code for the same amino acid. Okay, so that's why there's lots of mays in there. Okay, so it's a bit of a weird topic, this, because mutation is on both year 1 and year 2 of the specification. So this year we're going to learn some basics about mutation, and then next year we're going to introduce some other types of mutation, which means unusually this topic is actually on all of your exams potentially. So it can be on paper 1 with the material we cover this year, paper 2 with the material we cover next year, and of course it's a prime candidate for paper 3 because it's already synoptic. You learnt part of it in year 1 and part in year 2. Okay, so this year we're going to concentrate on 2 types of mutation. We're going to concentrate on substitution and deletion mutations. Okay, so substitution is where one base, or one nucleotide if you like, is exchanged for another during DNA replication. So the wrong nucleotide lines up in that space and is joined into the DNA molecule. It's just spontaneous events, accidental. Okay, or you can have a deletion, and this is where a whole nucleotide is just not joined together by the DNA polymerase into that long chain during DNA replication, and it results in the loss of a base. So we've got one less base within the gene. Okay, so let's have a look at the impact of these. Now you've got this in your keynotes with some examples, and then I'm going to show you some other examples as well. So substitution mutations, the first one we're going to look at. Okay, so if you have a think about the amino acid glycine, okay, so we just write this as GLY often in the exam questions. It's coded for by a whole host of triplets in the DNA. Okay, so you can see these are triplets rather than codons. This is the code in the DNA. So a substitution of the 3rd base of any of those triplets wouldn't actually alter the amino acid code it codes for. This is due to the degenerate nature of the code. Okay, not all base substitutions cause a change in the sequence of the encoded amino acids. So that's known as a silent mutation. Shh, a silent mutation. Okay, so there has been a mutation. The DNA base sequence has been altered, but it did not result in a change to the amino acid code. Okay, and that's because of the degenerate nature of the code. If you look to the code, often it's that last base within the code. You can change that one and it often doesn't result in a change in the amino acid that's coded for. A substitution of the first base though, so in this case CCT, changing one base at the beginning, changing that C, substituting it for a G, so that nucleotide containing the cytosine is substituted for one containing guanine at that position during DNA replication, resulting in a change to the amino acid, the base sequence, that will change the amino acid sequence, because that code in DNA, that triplicate in DNA, codes for arginine. So that is going to cause that change. Okay, so don't assume substitution mutations will not change the primary structure of the protein. Many of them do, because it's not the last base that's changing. When a mutation causes a triplet code for stop, that can have a really dramatic effect, because obviously the growing polypeptide chain stops way before it's supposed to. Okay, and if that were the case, then the protein probably wouldn't be able to perform its function, because it would be a shorter chain, wouldn't fold up in the same way. So at the bottom of the page, we've got an examiner's tip here. So the examiner commonly asks you to identify types of mutation. So you have to kind of look at the code and see what's happened to the code. Sometimes they present it in DNA, sometimes they present it in RNA. Okay. The follow-up questions are always about the impacts. There's a big strong link now to structure, the primary structure. If it does change, does that give you a change in tertiary structure? So it's really important that the depth to your answers includes your protein structure knowledge. So if you do say you've got an altered sequence of amino acids, you need to say this would result in bonds in different places between R groups, and so a different tertiary structure. And if it's an enzyme, you would be saying, and that would alter the shape, the structure of the active site. It would no longer fit the substrate, and so it wouldn't hydrolyze that reaction, or whatever that job of that enzyme is. Okay, so back into my PowerPoint. Okay, so we're going to take that original, which without the mutation was met pro loose sir. Okay, and we're going to mutate here. So we're going to imagine that during DNA replication, it's this codon here. Okay, so if we count 1, 2, the third codon, and it's the third base that's been affected, and that is the change. So we've changed from originally having a thymine on that strand to an adenine. Okay, so let's have a look what happens to the primary structure of the protein with this mutation. So it's a substitution mutation of the final base in the third codon, resulting now in UUU on the mRNA at that position. So we'll still code for the same amino acids at the other positions, but now that change of that one base has, in this case, resulted in a different amino acid. So instead of leucine, we've got phenylalanine. Okay, and that's because we've gone from UUA to UUU. So if we have a look at our table, we've gone from UUA to UUU at the top there. So we've actually changed amino acid. Okay. So this is an example then of a substitution mutation that could affect the function of this protein that this gene codes for. Now I say could because it's only changed one amino acid. But if that amino acid has a really important R group, so that leucine that would have been in that position was involved in holding a bond with another amino acid in that polypeptide chain that holds the tertiary structure. And now by changing to this other amino acid, we no longer have the right chemical group for that bond at that position. that bond won't be there and the tertiary structure will change. So if you have a tertiary structure change, that would affect the protein's function. Okay, right, so back into your keynotes for a moment. Okay, so we're moving on now onto page 12, which is this bit here, deletion mutations. Okay, so we've done substitution mutations. We're going to have a look now at deletion mutations. Okay, so this is the complete loss of a base in part of a gene. Okay, so the loss of a base in a region of DNA coding for a gene. A deleted nucleotide results, though, in an alteration of the triplet of the base triplets from that mutation on. Let's see this example here. Okay, so here at this position, this G excuse me, I've just had to pause the video because I've realised that this picture here in your keynotes is an addition mutation, and I'll talk to you about addition mutations in a moment. So do you want to just put a little bracket around it and put addition? Can you see this C here in red? That's actually been added. Okay, so I'm going to find a different example to talk through the rest of the deletions for you, and if you want to, you can copy that example from my PowerPoint and bob it in your keynotes here. But just for this one, okay, we'll put that one just to one side for a moment. Okay, so returning to my PowerPoint, because I want to give you a good example of a deletion mutation for your keynotes. Okay, noting that that picture was an addition, which I'll explain in a second. Okay, so deletion mutation, here's the original sequence in the DNA. So during DNA replication, this is what's happened. This nucleotide here containing thymine just was not included within the newly synthesized strand. And so it's been lost from the base sequence. Now, what's happening when that base is removed is that every single base downstream has to shift one place to the left. Okay, so you actually change the reading frame for every codon. Potentially every codon downstream changes. Okay, so these are the complementary RNA strands that would be produced during transcription. Okay, so that I can work out what amino acids the DNA codes for. I first transcribed it, and you can see that this transcript from the deleted gene, when that's transcribed, is one base shorter, and they've all moved to the left. Basically all moved one place to the left. Okay, so what impact does that have on the polypeptide chain? Well, we can start to have a look just even with this very short base sequence. Okay, so this is our original and this is our mutated. Okay, so if I've now, because I've looked up using the codon table. Okay, so you can do this alongside me. Okay, so AUU okay, so if you've looked these ones up, you can see that AUU was ILE, GAC in the codon table was ASP, and GUC was VEL. Okay, so let's have a look at the mutation then. Remember, the mutation happened here. Okay, so we really want to see what's happened to this codon and the codon downstream. Okay, so the first one's the same, but as soon as you start reading them from the point of the mutation, GCG gives us alanine, and then UCC gives us serine, so SER. So you can see that with a deletion mutation, the frame shift is to the left, and because of it, not only does the codon here change, every single codon downstream changes, resulting in many amino acids changing. So from the point of the mutation, the impact downstream is massive. Okay, so back in to your keynotes. Okay, so remember that diagram in your keynotes is showing a different kind of mutation. It's not a deletion mutation. Okay, but I'll come to that one in a second. Okay, so the deleted nucleotide, like the one we've shown in our picture here, that has caused an alteration in the base triplets from the mutation onwards. So because this is a deletion mutation I've shown you in the PowerPoint, you get a frame shift to the left. Okay, if this occurs quite early in the sequence of the gene, then you're basically changing all the triplets, and so you can alter all the amino acids downstream. A deletion that occurs towards the end will have less of an effect because you'll have less amino acids to alter, if you like. Okay, right, so we want to check our understanding of deletion mutation. Okay, so I've got a bit of a challenge for you. Okay, so if you imagine yourself a chain now, met, pro, lys, glu, trip, al. Okay, so it is 6 amino acids in length. I'm going to ask you to produce your diagrams to show the DNA code that will code for those amino acids. Different people will pick slightly different triplets, and that's okay. If you're not feeling very confident about this, that's fine. You can just actually let the video play and do the same one as me, if you like. That helps you understand it. But if you fancy a challenge, come up with the codes on your own. Your DNA code, your mRNA code, and obviously it should match your amino acid sequences. I want you to do a before and an after mutation. It's a deletion mutation I want you to do, and your single base mutation should occur on the middle base of the third codon. Okay, if you're not sure, that's okay. You can have a look at mine. I'm going to show you another example now that I've done on paper. But if you're feeling that you want to have a go, then have a go. Okay, so if you have had a go, brilliant, you can be able to compare yours. We might not have exactly the same DNA or amino acid, DNA RNA codes, because obviously it being a degenerate code, there's a bit of choice for some of these amino acids. They can be coded for by more than one codon. But we're just looking to see that you've done the right thing. If you're just working with me, that's okay. You just use this one. Okay, so that's my chosen code at the top. So I worked backwards from the amino acid sequence, and I picked from the table the codons that I liked to code for each of my amino acids. So that's in pink. Okay, then of course I've done a complementary DNA template strand in my blue. Okay, so that's my original on the top, and this is my third codon, and this is the middle base. So I know that's what I'm going to be deleting, that thymine there. Okay, so imagining that's happened, I've got a frame shift to the left because you can see where I've removed that thymine. Now my TC's moved into this codon, and that T's the beginning of the next codon. Okay, so we've got, start again, the cytosine's moved, and then that T is there at the beginning of the next codon. Okay, so what impact has it had? All right, so even if you've done this yourself and you've got a different DNA base sequence and a different RNA base sequence, the outcome should be the same. Okay, so where you've removed your base from the DNA, obviously it will be missing from the codon. So both in your DNA at this position here and in your codon, there's a letter missing. So there's an A missing here and a T missing here. They've been deleted. And everything else has moved to the left by 1 in a frame shift, which should result in a change downstream of amino acids. Okay, so you can see mine has yours. Okay, brilliant. That's what I want to show with our deletion mutation. Okay, so that diagram in your keynotes shows an addition. Okay, so I'll just come back to it. That's an error by me. Okay, I've obviously not been paying attention when I've popped that in. It is an example of a frame shift mutation, but if you look, it's actually an addition of a C there, and that's pushed everything to the right. Can you see that all those bases have moved to the right hand side? So this is an addition mutation. Now, addition mutations are only on the second year of our course, so they're only they're not something we're not going to talk about, but we didn't need to talk about it yet. But now I feel I need to explain it to you a little bit more. So addition mutations are literally the opposite of a deletion mutation. So we've got a base added to the base sequence for that gene, which causes a frame shift to the right this time. And in this example, you can see that that results in the 2 amino acids downstream of the mutation changing. Okay, so it shows the same. It's as dramatic as a deletion mutation is. The only difference is that the frame shift is to the right. So what I would do in my keynotes is I'd put a little box around that and say that this is an addition mutation. The frame shift in this one is to the right hand side. And then I would probably steal that slide and just add that into my keynotes or into my revision notes at that point. Okay, so back into the PowerPoint. Okay, right, so we do need to know a little bit more about causes of mutation. Okay, we have to be careful here because sometimes students, when they get to this section, think about mutations in a very negative way because we keep talking about them in the context of genetic disease. We do a lot in the exam questions. Here's a mutation. Isn't it terrible? What impact does it have? It stops the protein from working and causes this disease. Okay, but we need to also broaden out and link back to mutations when we think about them being beneficial sometimes as well. Okay, so in your keynotes, so carrying on from below where it said deletion, it's this section here we're having a look at now. Okay, so mutations. So mutagenic agents are things that increase the chance of one of these mutations. Could be substitution, deletion, addition, those events where a nucleotide is missing, added, or substituted during DNA replication. That can be increased, the chance of that happening can be increased if you add a mutagenic agent. Okay, so if you're wondering what kind of things are mutagenic agents, any carcinogen, anything you've learned to be causing cancer is likely to be a mutagenic agent because cancers are the result of mutation. Okay, but remember they're spontaneous too. They're natural and random events, but they do cause permanent changes to the DNA that is then passed on to future generations. They do occur at set rates, which is different for different species. So we do use them, and we can talk about them when we're looking at studying organisms and how much we change over time, because we can use our mutation rates as kind of like a timer to see how long it's been since we shared common ancestors. Okay, so mutations are responsible for genetic diversity in populations. We need that. We're going to talk about that a lot. We talk about natural selection later on this year, and then we're going to talk about natural selection and speciation, that's the production of new species, next year. And we need mutation for both of those processes. They do, however, often produce organisms that are less well adapted to their environment and can alter cellular activities. So, we can put a positive spin on mutations, but often they are negative in their effect. Mutations that happen in gametes can prove fatal, and mutagenic agents, the things that increase the rate of mutation above that of the natural sort of spontaneous events that fuel natural selection, are things that include high energy radiation, X-rays and gamma rays, which damage the DNA molecule and chemicals that alter the DNA structure and interfere with the DNA replication. So, named chemicals. Often the examiner will link this to DNA replication in the exam question. Okay, so moving on to our exam questions, we're going to have a go at some of these mutation questions. So it's question 5 in your question pack. Okay, so question 5A, gene mutations occur naturally. Give one factor that increases the rate of mutation. So like we just said, you need to just name one thing that we know increases the rate of, or the risk of mutations happening. So it's really important in your answers here that you either state ionizing radiation or high energy radiation or high energy particles, or give an example, so X-rays, gamma radiation, or a named mutagenic agent, something like benzene. Okay, so just learn maybe 2 different ones and keep using those. You'll see the common ones come up on the mark scheme. Okay, 2 that you can remember well. The table shows the DNA-based sequences that code for 3 amino acids. Some substitution mutations would affect the sequence of amino acids in the polypeptide chain, and others would not. Use the information in the table to explain why. Okay, so if you look at the table, you've got a couple of amino acids there that have more than one triplet that codes for them. So you can create substitution mutations, particularly this one here. If you substitute the last base in that triplet, it wouldn't matter. You can substitute it for any other and it still codes for the same amino acid. So that's the one I would go for, and I would use that. And in terms of the structure of your answer, you're going to get mark points for saying glycine. You can substitute the 3rd base for anything else and you'll still get glycine. Okay. So we've said why some wouldn't affect. Okay. So now we need to say those that would. So then if we pick this one here, isoleucine, and we take the last base and we change it to a C, we know that that substitution is going to change the base, the amino acid that is coded for. Okay, so in the mark scheme it's here. Okay, so isoleucine, if you substitute no, it's not that one, it's there. If you substitute C for any other, they've done it the other way around, you get isoleucine from instead of met. Okay, but because it's equivalent, we get the mark point there as well. Okay, so we've said 2 things, but we've got 3 mark points that we want to get. Okay, so we've said for glycine we could change any of the last base and it wouldn't affect the amino acid coded for, so we could substitute the last base for any other and it still codes for the same. We've said for isoleucine, if we substitute the last base for C, you would get met, or we can do it the other way around. So we could say met, if you substitute that C for any other base, you end up with isoleucine. So there are 2 mark points worth of information. Okay. So we still need one more different thing because it's 3 marks and we're still using information. Okay, only the information in the table. So now if we have a look at what else we'd need in there. Okay, so you can use them for the 3rd example, substitution of either of the first 2 bases to give you a different amino acid. Okay. So in any example, if you like, if you pick, for example, that one here, CC was substituted for a TA at the beginning, then you change from that one to that one. So it's just illustrating another substitution that could happen. We've only really talked about single base mutation. So it is possible sometimes that you get multiple substitutions, so you can substitute 2 bases at the same time. Okay. So the final part is describe the role of nucleic acids in the synthesis of polypeptide, and everyone should be able to pause and do this one with no help at all now. So just consolidating. Okay, so join me back for the answer. Mark scheme. So sequence of bases in the gene determines the amino acid sequence, so like the overall idea. A description of transcription of the base sequence of the gene into mRNA, and that the mRNA is split up into codons. Each codon coding for one amino acid, I would add in there. Then you would have some sort of reference to base pairing, as it could be how that transcription happens, so how that transcript forms. On this mark scheme given in terms of letters, but on the new mark schemes you would have to have the full names of the bases. And then the idea that a specific tRNA will carry an amino acid during translation. It has an anticodon which is complementary to the codon on the mRNA. That process is happening at the ribosomes, and the ribosome, in terms of the final mark point, is the site of translation. It moves along the mRNA to include the next codon until the process stops, the stop codon is reached. Okay, so it's a weird thing with the mark schemes on these, that there's a lot of information you would write. Your answer's probably about half a side of A4, and then they just look for little snippets within it. The reason your answer has to be a side of A4 is because if you wrote this, it doesn't make any sense as an answer. This is just the highlights that they're looking for within your answer, the bits of detail and depth they would expect if it was a good answer. Okay, right, moving on to question 6 in your question pack then. Okay, so first of all, what is the role of RNA polymerase in transcription? Got to be careful with this one. I would be really detailed. Okay, so if you have a look at what they've got in the mark scheme, I would say joins adjacent RNA nucleotides together between the sugar and the phosphate to form the sugar phosphate backbone, or by condensation reaction producing phosphodiester bonds between the sugar and the phosphate. So be as descriptive as you can in its role. Name the organelle involved in translation. It's our ribosome or rough endoplasmic reticulum. Okay, so then we've got a picture. It's got the base sequence here of the mRNA. So what it's asking you for, first of all, is the DNA from which it was transcribed, and then it wants the anticodons. So I'll stop the video and have a go. Okay, so show you the answers. All right, so do you want to check your answers against those? Okay, now the fun bit. The table shows the mRNA codons for 3 amino acids. Name the type of mutation represented by mutation 1. Okay, so if we're doing a comparison to the original, we can see that here is where we have a change, but downstream does not appear at all affected. So this is a swapping of the U instead, isn't it? So that's a substitution mutation, and that's the common thing that you'll be asked to do. Okay, to look at these and to see what kind of mutation. So always look for the change, and then is that the only change, or has there been a frame shift? So that in this case there hasn't been any frame shift. That's one base change, and so it says substitution. Use the information in the table to identify amino acid X. So you have to go back to the other page. We need the codon, okay, for X. So this is X, this is the codon, GCA. So GCA. Okay. It's quite common for students to look at the wrong line, maybe look at the anticodon for that one and pick the wrong amino acid, so just be careful. Explain for part 2 then. Explain how each mutation may affect the polypeptide for which this section of DNA is part of the code. So mutation 1, we need to have a look at what impact it's had. So did changing that base change the amino acid that's coded for? In this case, mutation 1, we're still coding for the same amino acid. All right, so no change to the amino acid sequence. The code is still for alanine. It's a degenerate code. The same amino acid has been coded for. Okay, it's really common for them to give you 2 mutations, one with an impact and one without. So we're kind of expecting now that number 2 is going to be a different story. So remember to compare mutation 2 to your original mRNA. The first 3 bases are the same, and the second 3 bases are the same. And then it's the third codon. We can see that this has changed. So it's gone from G, U, U to G, C, U. So this would change the code. Now at this point, everybody wants to know what kind of mutation it is. When I do this in class, everyone's like, I don't know how you tell whether it's a substitution or whether it's a deletion. And I say, that's not the question, so don't worry about it. So it could have been a substitution, okay, because you could have just swapped that U and C, or I suppose it could be addition, so that could be the addition of the C and then shifting to the right. The U's moved 1 over to the right. Doesn't matter, because that's not part of the question in this case. They're just asking what the impact is. So mutation 2, there is a change. Valine will be replaced by alanine, and that will have an impact. So the second part of the question, really the second mark point, is for saying what will happen to the polypeptide chain. So really you've got to talk about how it will fold up into a different shape, but you've got to go a little bit further than just shape, because that's a bit too GCSE now. So we use the key term tertiary structure, and bonds are very important. So if you talk about the position of the bonds that hold the tertiary structure being in different places, so it folds up into a different overall tertiary structure, that would be a better answer. Okay, so a bit different now on 7. All right, so what I've done is I've answered the question as a student. Okay, so I want you to have a look at the student response. Okay, you might have to look at the PowerPoint if it's not showing up very clearly on the video, and I want you to mark it and improve it. Okay, so pause the video now and do that task, and then I'm going to start going through. Okay, so you should have done the task by now. Okay, you're rejoining me in the video, so you've marked it. How many marks did you give it? Okay, I'm going to tell you how many marks I gave it. 1 out of 7. Okay, so let's have a look at the answer and why it's only got 1 out of 7. It didn't appear too bad at first, but as you look at the detail, you're going to see why. Okay, so the first mark point they didn't get. Okay, it's no good. You cannot name one mutagenic agent and say smoking or cigarette smoke or something vague like that. It has to be a specific name. So if you're going to go for something chemically, okay, so a carcinogen, you have to name it. So you have to write benzene, or you can write X-ray, or you can write gamma radiation. So it's got to be specific. Okay, in the second part of the question, the students only given very vague detail. Okay, so they were asked to describe how the deletion mutation altered the structure of the gene, and they've just said it's changed the base sequence basically, and that's not enough detail. So there's been a removal of a nucleotide during DNA replication, resulting in the loss of a base in the sequence. This causes changes downstream too. Because it's a deletion, there's a frame shift to the left. Can you see the difference in the detail level there? So that's what has to be there to upgrade their answer. Looking at the second part of the question, okay, describe and explain how the altered gene could result in the flax plant having white coloured flowers. They have got 1 mark point in their answer here, because they said that there would be a different amino acid sequence, but that's it. Everything else is at GCSE level. There's no detail or depth. So to improve it, they would have to say the deletion in the gene would cause a change in the bases in the mRNA. So when that was translated, it would result in a different amino acid sequence in the polypeptide chain. Then they needed to go on to tertiary structure here. So it's no good saying so it won't fold up in the same way and be the right shape. It's all too GCSE. So here we're going to say this would change the tertiary structure because bonds between R groups would change position, they'd be changed, resulting in a change in the active site structure, because this is an enzyme, remember. Not work, it won't work, was too vague. You need to use the information given to you in the question. So if you do that, then you would go on now to say that if this enzyme's active site was not I didn't have the right shape, the white pigment would not bind into the active site and the lilac pigment would not be produced. Therefore, they would have white coloured flowers. Okay. Right, so there is an extension question in the PowerPoint for you to do some more practice, if you like. I've popped my answer in there as well. Okay, so you can have a go at that. And then to finish off, I've got a bit of a suggest challenge for you now. Okay, so if you want some extra practice, extension questions first. Okay, right, so suggest 5 degrees of separation. Can you link these two items together in 5 steps? So you put 3 steps between them. A mutation in the gene for lactase and someone being lactose intolerant. So stop the video and have a go on paper. Okay, right, so this is what it should look like. So if you've got a mutation in the gene for lactase, we've got a change in the base sequence of that gene. Second one, this would be your linking one. This would mean the mRNA codons would be different, so code for different amino acid chain. 3, the lactase's tertiary structure therefore would be different. 4, lactose would not be able to bind into lactase's active site, so the lactose would not be hydrolyzed to glucose and galactose, and that would result in lactose intolerance. And for anyone who knows anyone with that lactose intolerance, they can tell you that you know, the not being able to hydrolyze that disaccharide, if they then eat lactose, the symptoms are like diarrhea, because obviously the water is drawn into the lumen because you can't absorb the disaccharide. It stays in the lumen, lowering the water potential and bringing water out. And also wind, which is not very pleasant, because the lactose is used by the bacteria in the large intestine. They use it as a respiratory substrate and they produce gas, which causes bloating in the intestine as well. There you go. Right, bit of synoptic there. So that is that for this part of the topic. Okay.